Hi everyone. Welcome to today's webinar. Yes, my name is Alex. I am a staff scientist bioinformatician at NYU Langone Health in the Cardiovascular Research Center. And today I'm excited to bring you a talk which is sponsored by Biotech XYZ. This is a local life science networking group here in New York City. Biotech XYZ is a 501c3 certified nonprofit organization founded in fall 2022 by executive MBA students at Cornell Johnson. It now brings together students, research trainees, and academic and industry professionals in the New York City area. Biotech XYZ organizes and co-sponsors a lot of very interesting events, including an upcoming panel on biotech venture capital investment planned for April 9th. So please be sure to follow them on LinkedIn to stay up to date with their events, or if you're interested in volunteering and organizing your own. So today it's definitely my big honor to introduce Dr. Parashar Dhapola. And before getting started, let me just say we will have time for Q&A at the end, and I'll unmute the mics at that point. But please feel free to write any questions in the chat in the meantime. So Dr. Parashar Dhapola is co-founder and CEO of Nygen Analytics. Let me pin here. There we go. Dr. Parashar Dhapola is co-founder and CEO of Nygen Analytics, a Swedish tech bio company building AI-powered, cloud-based analytics platforms for single-cell genomics. Dr. Dhapola completed his PhD in computational biology at Lund University in 2022. His doctoral research at the Lund Stem Cell Center focused on developing computational methods for single-cell transcriptomics and hematopoietic cell state estimation. Parashar is first author of SCARF, a memory-efficient single-cell analysis toolkit published in Nature Communications in 2022. SCARF enables atlas-scale genomic analysis on standard laptops and currently serves as the foundation of Nygen's commercial platform. Earlier, he developed Quadbase 2, published in Nucleic Acids Research while at the CSIR Institute of Genomics and Integrative Biology in India, where he held the prestigious SPM Research Fellowship. Parashar's research expertise includes single-cell multi-omics, machine learning, hematopoiesis, and scalable computational genomics methods, with applications in precision oncology and cell therapy development. So, Parashar, we're very excited to have you here today, excited to hear about your journey, the story of Nygen Analytics, as well as to learn about your hard work building CyteType. So with that, let me go ahead and stop sharing. And Parashar, please take it away. Do you have any slides or want to?
Yeah, perfect. Thanks a lot, Alexander. I do have slides that are focused on the product, but I can start with a bit of maybe an introduction and a background on myself as well, especially in context of what we do here at Nygen. Let's see if I can bring my screen up real quick here. And hopefully I have the right screen up. It doesn't show me the usual border on my screen, but I guess you can see my
Yeah,
full
I see a
screen.
slide that says Nygen, evidence-based cell annotation.
Perfect.
Okay, perfect.
Perfect. Yeah. A little quick background on Nygen, who we are. So thanks a lot for the introduction, Alexander. And as you mentioned, right? So Nygen has been focused on building, using AI and the current advancements in LLMs to better understand biology behind these large scale omics data. But let me backtrack a little bit and start with the journey. So I finished my PhD in 2022, and we immediately started sort of established Nygen as a startup with an idea that we can facilitate and add value to whatever academia is doing in the space, which is a lot. You have these top labs around the world, in Broad Institute and in ARC Institute in the US, which are really becoming the hubs for producing a lot of the single cell data and methods associated with them. And so we were thinking, okay, where can a small, scrappy startup based out of Lund like contribute to this field and attempt to push the field forward? And and we had our own thesis of what we can do about single cell data and other growing omics technologies. So the thesis for us has been very simple so far, is that the current methods and how people work and develop new methods is very sort of from, let's say, PhD researchers. It's a lot based on developing new methods, developing new algorithms, but connecting them with pharma is a long journey. There's a lot of sort of infrastructure problems you have to solve, biology decoding that you have to do for these people, and then you have to adopt all of these methods to people's workflow. So in pharma, for example, people don't just run Cell Ranger and Seurat, and that's it. That's the end of the day once you have done your cell annotation. But that has to fit into different needs of different people. It could be discovering new targets, or how do we do cell therapy, and all of those questions could be quite involved. And then every pharma has different questions towards that. So that's what sort of, with sort of that thesis and aim, we built Nygen. And of course, towards that big aim, there's a lot of stuff that needs to be solved along the way. And one needs to understand, like, you as a startup, what do you offer that's unique? So for Nygen, that has been, of course, to start off building on Scarf, which was our first sort of technology, sort of a backbone of the startup, is to run a lot of this data analysis on the cloud in a really scalable manner on tens of millions of cells. You can run it on single digit gigabytes of RAM. And we orchestrated all of that in the cloud. And some of the pharma companies have been using this in a brute force way to, how about we run different instances of highly variable genes and clustering resolutions, and then just fan out those analysis on the cloud simultaneously. So we enable that kind of groups that are interested in doing those kind of research, those kind of experiments in brute force, and then we provide sort of infrastructure for those uses. A lot of this is not visible through our public channels, where we kind of have put that technology in a GUI and enable, let's say, non-technical wet lab people to be able to do data analysis themselves. So that is one part of what we do in terms of enabling analysis by scalable infrastructure. And the other part that we have been focusing a lot more in past 1 year and so is, how do we reveal more biology from the single cell data? And the one problem that we're laser focused on right now is cell annotation, and CyteType is an attempt in this direction that I would be really happy to talk about in my uh, later slides. But going back to the startup again, is that we are when that once coming out of academia, and that's what I learned in the initial years as well. Is like, you are a little bit fallen in love with the solutions you have developed, and not really connecting with the real problems in the what that are out there in the wild, in the pharma companies and biotech companies. And that's truth seeking is something that, as a startup company, that becomes most valuable to anybody. And in today's age, where everybody has Cloud Code and other tools and AI in their hands. So building is no more, as they say in the Silicon Valley and Y Combinator, building is no more the moat. Understanding problems deeply and connecting to people is the real challenge. And people can over weekends build pretty solid sort of at least the MVPs of solution. They might not be secure, they might not be scalable, but they give you a sense of solving the problem. So this is why we have been doing a lot of that work, connecting a lot with pharma companies, and any of you bioinformaticians and other people in the crowd who are sort of already involved and thinking about how can they sort of go in this direction. My only advice would be that don't start with building like I did, because I had like already started with a solution, and like follow and like go out and talk to a lot of pharma companies. It's worth it to understand the problem deeply. You can never understand problem enough, because the more you talk to the people, the more you learn from them. And it took me a long time to stop pitching the solution and understanding the problem. So that was the only advice, and probably something that's already well known, but very hard to implement when you're so close to building the solution. And that's something that I have personally struggled with a lot, at least initially, and continue to do it to some degree. Of course, that's because we are scientists. We love to develop solutions and then talk about those solutions. The other aspect is, of course, people, when they think about startups, they think about funding, they think about how to fundraise, and that game has also changed a lot now. Because you can build things very quickly, you can put it in front of people. So a lot of different sort of equations are changing there. If building is not the moat, what is? And that is, of course, learning the problem, but also a lot of people say that you have to develop some sort of asset. So for us as bioinformaticians, because most, if you want to build something, it's best to build something in open, give people access to it. Not always feasible when you're a for-profit company, but one should try at least because that's how you best engage the community, and that's how you sort of jumpstart your products out in the market. The other part is that you have to build some IP, and that's why you raise money for, not to build your tools, but to build some sort of IP. And the IP could be maybe, for example, in our case now, what we are doing with CyteType, and I'll probably not talk so much about it today, but maybe I leave some sort of crumbs along the ways that we are using CyteType to process very large scale data sets for different company, and whatever the outcome of that CyteType is being used to now train certain models downstream. So CyteType becomes a way to create IP for us that would be valued by investors and other people down the line. So that is another point that one has to think about when building startups, is to think about how are they going to deploy these tools to create IP. So tools them as a means to an end, but then the tool itself should also build something unique and position itself in a way that solves the problem in a new way. So just those two advices, I would say, unprovoked, given that Alexander mentioned, maybe if I could talk a little bit about my journey, is that would be to focus on the problem and then to think about why would you need the money? And in that case, if it's you're doing massive computation on a scale which will differentiate, then yes, you raise money for doing that, but not to build your tools, okay, because that has become, yeah, you can do that very easily at very low amounts. And things used to be very different 5 years ago, like when we started, when you would raise money to hire engineers and build your first products out and in a nice way, but you don't raise money for that anymore. So that is two things. But again, here being in Europe, how you raise money, how the market looks like from that perspective is very different from what it is in the US, where it's more aggressive, more all in, and I would say more exciting sometimes as well. That I hear about from my other sort of founder friends on the state side. Yep. With that, I'll probably now switch gears and talk more about why probably most of you are here for, is to a little bit learn about CyteType. Uh, so CyteType, as the title says here, it's an evidence-based cell annotation system. But the underlying sort of philosophy that I'd like to leave with all of you today is that it's a biology. Not a is biology a classification problem, or is it not a classification problem? Because when we think about cell annotation, we think about, okay, labels that we will put on every cluster in the single cell data. But is that annotation, is that enough, or is there much to beyond that towards in the annotation process itself that needs to be revealed? And should this be approached from classification perspective, that you just have to identify what those labels are, and those labels are some sort of a ground truth that just needs to be revealed behind those expression vectors. So I will talk a little bit about that and from a philosophical point of view as well along my slides, but let's dive in here. Alex, anything you'd like me to talk more about on the startup journey side, or if you have any questions in mind?
I think that's absolutely fantastic. We have a couple questions, but I think it's best to keep the Q&A towards the end. So yeah, really appreciate that. Really, really helpful, useful insights. Thanks.
Absolutely. Great. Cell annotation is a hard process, continues to be even in this time and age. Most of this is because, that your accuracy will drop instantaneously if you move towards new disease context. The reality is that if you're working with, let's say, human samples with patients and disease context, there's a lot of drift that happens there. People have different genotypes, they have different treatment modalities, there are different advanced stages of their diseases. And you can never really have a gold standard reference when you're working with, let's say, clinical samples and real world samples out there. So your accuracy, what you do with your healthy references in a very curated study, that doesn't translate very well to in the real world data sets out there. And you will have that sort of classifiers will break in that reality, and then you will see drop in your annotation accuracy out there. If you would bring in experts, creme de la creme people who understand the biology, who are cell biologists, and disease biologists, pathologists, they will still have a lot of variability in how they annotate manually things. Because when you get in a deep enough resolution of a subtype or a cell state, people have different opinions on how they intrinsically are weighing different features, different genes, what's present and what's not present in your data. And that naturally brings a lot of variability in annotation process. This is something these numbers are like have been picked from a couple of reviews articles that are out there. If you search for like the annotation and what's wrong with them out there. And I'm just sort of contextualizing this for you here. And then, of course, at the end, you have the rare cell types that are missed in most of the cases. This is partially outcome of your clustering and upstream steps themselves. That if you removed all the highly variable genes that were part of that rare cell types, then of course you will not reveal them. If you don't have clustering at enough resolution, then of course you'll miss them. But even when you have them, can you distinctly identify them, or you'll mislabel them, and which is also happens because the rare cells are highly imbalanced and you don't have enough of them in the data, and your features are not weighted properly. So that is also something that happens a lot more in a lot of studies, that you would miss them, and all of these combined make annotation a pretty hard process overall. But it's a little bit of an educational slide for people who are a little bit not as initiated, but I guess most of you are a little bit aware of some names here. But just a way of like segmenting all the hundreds of tools that are out there actually to help you with the annotation process and with single cell data, and we can broadly define them, categorize them into four groups, starting with the reference-based methods. These are methods of choice, for example, in pharma companies. They love it because, it brings a lot of predictability. There's something you can document, audit easily, and you can build your own gold sort of reference, gold standard reference, and then you keep using these tools downstream of those. So they're fast, they work well when you have well curated references, but in real world data set, they almost always break. And many of people have questions, do you need all these very specialized methods to be able to do reference-based annotation? People also call them transfer learning-based annotations. When even very simple machine learning tools like support vector machines can become a very strong contender and outperform many of these when trained on the right data. So idea being here that your basic classification tools trained on curated references can perform really well and fast. But of course, when you have disease cases and real world data, they tend to drop a lot in accuracy. But this is something which is most used today out there. There are marker-based tools like Garnet, CellAssign. They leverage known marker databases, but they ignore a lot of biology because marker databases don't capture all the biology of cells that is highly nuanced, and you do tend to then sort of focus your attention too much on certain features and often misannotate. So we often see without any context, wrong cell types coming up in the context where they should not be, because let's say in cancer they start expressing some surfactant genes which are usually in liver, but those cancers do that, then those cancers will start being marked as liver cells, and I'll show you an example of those. And that happens a lot because those surfactant genes are not present anywhere else usually. So they can highly sort of get biased and anchor biased, as one can call them, towards certain markers, and usually produce a lot of artifacts, especially when you have disease biology in your samples going on in some sort of disease context. Transfer-based methods, these are heavyweights, very exciting new approaches that have come out in the last couple of years. SCGPD was the first one, but then there are more and more different approaches. You have universal cell embedding for across species data set, something that a lot of pharma companies, especially translational teams that are taking studies from mouse to humans, are very interested in looking at and have been poking around with these kind of methods, but also like methods that are more coming from like LLM domain, like cell to sentence from Yale. And these people have also been like sort of been adopting that for annotation purposes as well. But these methods are very black box methods. There is some sort of drift and stochastic nature to these methods as well. So they often don't produce such great results as people have over and over benchmarked. But they are promising. They're going in the right direction, but we don't have great tools that would replace, let's say, a good reference-based tool today. That would probably be more exciting to use. And also, they don't give you a sense of causation and why, and those are of course missing. But that is also true for other methods as well, except for marker, where you can say, okay, because these markers are there, then that's a cell type. And at the end, you have the LLM-based methods like GPT CyteType that came out in Nature Biotech a couple of years in 2024, in autumn then, and has been very exciting. And then that you can just ask LLMs what the cell types is, and then it can tell you what it is. But the current implementations are very shallow. They're just prompting the system with like one or two prompts, and I'll tell a little bit more about this. But what's exciting about LLMs is that they can contextualize. They can put your data as, okay, where did this data come from? Which technologies were used? What was the assay used? And what's the background of the study? What kind of samples are we talking about? How these cells are sorted, and all that context can be baked in into the annotation process. And that's the promise of these LLM based tools. But then again, how do you build it in the right way? That's the big question. And that is basically where we start asking ourselves, can we add something over where these LLM based tools are leaving or the ball down? And that's where we picked that's where we sort of introduce CyteType. So CyteType is an evidence-driven, multi-step AI workflow specifically for single-cell annotation. Since the release, we published a preprint in bioRxiv in November, and then we have been getting a lot of interest from academic community to use CyteType, and some of these GitHub stars are indicating. And so far, more than almost 50,000 clusters, I can say, have been analyzed across 2,500 data sets by the people using it in public domain, and we are of course using it internally to also process massive amount of data. And I'll come to it at the end as to the LLM infrastructure and how do you actually make it work on this large scale without failing is again a massive engineering challenge that we have been solving behind the scenes. But let's talk more about like the core architecture of CyteType. And there are three things to think about when we're talking about site types. That one is that site type is contextualizing your data sets. What are the priors? What's the experimental context? Which are the enriched gene signatures, for example? That it is hypothesis driven. It will try to come up with different ideas. Okay, can it be this? Why should be this? And why it should not be that? And all of that. And it does that by live querying your expression profile. So going beyond marker genes, but looking at all your genes and okay, and asking like, what does this gene do here? And what does that gene do? And there, the hypothesis that we have internally is the thesis we have is that most machine learning tools today, they consider all your features, all your genes to be independent and identical. The basic statistical assumption of being IID. So and that's not true for genes. One gene's presence masks the effect of what other gene might be doing. And how do you really model that? How you weigh genes? How do you sort of model their epistatic effect. And this has been a big discussion in the field. It's really hard to do. I mean, that's the whole concept behind systems biology and how to do that. But we haven't had good models to do that. LLMs can do this on a smaller scale. They can give you like smoking gun hypothesis where certain presence of certain genes just mask away the effect of other genes because that's how the pathways are sort of built, and they can reason about that. So that is something that we have been doing here in CyteType, like building these hypotheses and testing them based on presence and absence of different genes, and having a biological reasoning behind those, and then validating that against you have to fight against hallucinations and all that process. So you have a validation step linked to all of that, where you have other agents and other LLMs that are sort of looking into it, doing a structured review of the process, connecting to external databases, external literature, saying does that make sense based on what we already know, or is it something that the LLMs are just cooking up on the fly and hallucinating? So all of that combined together with a good context, thorough hypothesis search, and validation allows you to really leverage these LLMs in a very, very sort of strong way to get high quality and nuanced annotations. So just probably this being the last slide, to give you like a little bit positioning of site type. So how are we addressing, what the challenges and limitations that LLMs today present? One is, of course, top marker genes that's being used by most methods today. We don't do that. We look at full expression profile, enriched signatures, looking at pathways. So we convert that data into more sort of a downstream derived data, like, enriched pathways and all of that. People don't usually do it because then you have to manage context. You don't want to like just overflow your LLMs with all this information beforehand. So then that becomes an engineering challenge that we solved, and I'll tell you a little bit more about that as well. With other LLMs, you're looking at their static pre-training knowledge. Whatever the LLMs have been trained on, they're just querying the data on that information. There's no live database integration search. It's not going on databases and checking, okay, does this exist in this database? Do I get that validation from some other known database as well? So this is where CyteType is doing that and not just relying on the static pre-training knowledge. as I told you a little bit about, and I'll tell you a little bit more about how we build like virtual expert panels to do validation, to do review of the process, how we are building, confidence scores so that you can, okay, is this is what they're saying is how confident is LLM about this? So currently there's no uncertainty metric, and which is also actually absent in many ML-based tools today, except a few which are more probabilistic, like Celesine. It provide gives you a full probabilistic sort of certainty behind every label it will suggest. LLMs in this case don't usually have that, and that needs to be baked in. So we built a whole way to have, confidence calibration with evidence when the LLMs say, and then we have benchmarked, does this confidence is meaningful, or you would expect when the confidence is low, it's often wrong, and when the confidence is high, it's often correct. So those kind of benchmarking we have done, and I'll talk a little bit about those as well. And then, if you're just asking, here's my marker genes, what is the cell? The LLMs can hallucinate, and that's something that we are sort of really fighting with CyteType. But one thing I'd also want to talk about, and something that most people don't talk about, is hallucinations might not be the biggest problem here. The biggest problem might be cherry picking, because when you're asking LLM something, they look at 50 genes, they might just take and talk about five genes out of them and say, okay, because of these five genes, it has to be this. So that's cherry picking your data. It's just ignoring everything else. That's not strictly hallucination because it's what it's saying is true, but not in the full context of whatever else you have. So inside that, we have been, developing those multi-step, well, let's look at all the genes. How does this gene contribute? How does this gene contribute? So it's a very exhaustive search. And then it becomes an engineering challenge that we solve in CyteType as well. But hallucination and cherry picking, these are the two things we really fight against, what LLMs are very susceptible to, and then you have to build an architecture to contain that problem. Skip this slide. So let's talk a little bit about where does CyteType enter the usual flow of a bioinformatician. You probably get, your wet lab colleagues will be working with, heterogeneous samples, that's a tumor microenvironment, and you run a basic data analysis, a secondary analysis maybe on SCVI or Seurat or ScanPy, starting with your raw count matrices, filtering out cells for the low quality cells, identifying highly variable genes in your data, running principal component analysis or SCVI embedding, so some sort of latent embedding of your data, which then you use to build a KNN graph of your cells, and then at the end you build sort of do a clustering and UMAP visualization of your cells. And then once you have these clusters, that's where you want to find out what's happening here, what's the biology behind these clusters, and that's when CyteType enters. So the packages CyteType R will be able to work with your AND data and Seurat objects and be able to pick this information and start its work from down there. So the first step, contextualization is really important, and that's done on two levels. One is on the study level, where you try to understand, okay, which pathways are generally enriched, what tissues am I looking at? User might not have provided you sort of explicit information about that. Users have ability to provide that explicit information, but then you can also derive that information, especially like this is important with clinical samples where you're taking histopathology, sort of, or biopsy samples, and then you have a lot of tissue and contaminating tissue from other places. And if you have processed the whole sample as is, then you probably you don't know that beforehand as an experimentalist or as a clinical sort of pathologist. So CyteType can come in and also do that tissue context identification as to what tissues am I looking at here by looking at highly expressed genes, by looking them in GTEx and other databases, and trying to build a study context. Then it goes cluster by cluster and builds a cluster context as well. Okay, am I seeing a certain kind of batches really overrepresented in this cluster or treatment? Some of the cells have come from certain treatment modalities. What's the disease representation in that cluster? Does it come from disease samples, normal samples, and all that to build sort of a context of a cluster vis-a-vis other clusters as well? And then we pseudobulk the whole data to be able to go in and check expression of any gene that we need to, and build it out into a database that is really, really fast for these agents to query. Because agents will be doing tens of thousands of such queries across clusters. So you really need that efficient packaging of your data and make it accessible to agents downstream. And that's where now the LLMs and packaged with as agents they come in, and what they will do, they will start building hypotheses of what those cells could be based on the study and cluster context, and start sort of collecting evidence from the database as to which genes are high, which are low, which are absent, what about this, what about that. So they do all these loops of asking questions and validating, and then compressing all that information into sort of what could be the right annotation based on the evidence that it's looking across different hypotheses. And this happens independently for each cluster. There comes a point later on in the pipeline where we sort of combine and build neighborhood awareness between these clusters and refine those annotations by looking at differential expressed genes between nearby clusters. So that's a refinement step of the hypothesis that happens after we have done hypothesis generation and selection on each cluster individually. But what also happens in this step is the virtual panel expert. So for every cluster, based on the cluster context, we will assemble a virtual panel of experts, up to 5 experts who will be looking at different aspect. It could be an immunologist, some sort of a disease biologist, and these will be sort of generated on the fly based on the cluster context that's relevant. And then they will go out there and sort of what we will do is sort of go on PubMed based on the keywords, the genes that we're seeing, and from every disease, every one of these experts will create a lot of query terms and fetch a lot of literature. I think what we're doing right now is up to 1,000 papers, and based on these keywords, and will try to enrich itself, live train as to what is the literature saying about these kind of cells out there. So each one of them will build its own queries. And will go out and run those queries on PubMed and collect a lot of literature. This is a way to get most updated information, but also, you don't have that hallucination baked in. Once it is trained, I know these reviewers have sort of updated their knowledge. They will then go and look at your data and start asking about more evidences, and will say, does this hypothesis make sense? Why doesn't it make sense? And in that process, they also connect with also not just literature, but also market databases as well. So once they have all provided their, feedback on this, what are the strengths, what are the weaknesses, then we feed that into a confidence calibration score. Like, are these reviewers converging? Are they diverging? Are they like they all have similar sort of concerns about the annotation? Then of course your confidence will low. And if the confidence is really low and it's actionable by these experts, then we will do a new sort of re-annotation with those updated hypotheses and updated priors. And at the end, what you get is like a cell type labels, of course, and confidence scores for each of your cluster. But the most important thing is that this annotation happens at multiple levels. You go from like really basic broad cell types to really, really granular and nuanced cell states. And these will be able to, as I show you in the following slides, that you get all this data at the end of your pipeline here. So most importantly, that's a lot of mumbo jumbo happening with LLMs. Does it work? And that's where a lot of our effort went into sort of benchmarking this. And we developed a companion package actually to our setup to be able to run this kind of benchmarking. So I'll talk really briefly about that as well. So first of all, we took these very large scale atlases for benchmarking, comparing CyteType, using different LLMs as the brains of CyteType, and then comparing that against single R cell type as GPT cell type. We use the author annotations as ground truth because that's the best ground truth you usually have in these kind of data sets, and then predict the predicted annotations from each one of these 4 pipelines as well. And then we do the similarity score calculation between these two. And that's a very hard problem actually, that how do you compare labels, because these are at the end of the day just text and characters. How do you say like if how close is CD4 positive cells to CD8 positive cells, or CD4 positive naive are close to CD4 positive memory cells? So all of that is sort of a very hard problem to solve. And people have traditionally said that you could use, cell ontologies and their hierarchy, but that is actually quite a not well-built hierarchy on cell annotations. So that's why we came up with a new solution, and we built a companion sort of a benchmarking tool called CyteOnto here. We do use the cell ontology terms, but we don't use them as is. We enrich these cell ontology terms in a structured process, embed them into a model, a text model, and in that embedded space, we run comparisons between predicted annotations and author annotations. So you would have thousands of these cell ontology terms in this sort of embeddings of these, and we will run embeddings of predicted and author annotations, and then do search. Okay, where did the author annotation hit? Where did predicted annotation hit? And what's the delta between those two? And that will give you a sense of a very continuous difference between how far is the predicted annotation from author annotations. Uh, so I will just run through this really, really quickly. Again, is that your input is your predicted annotation, author annotations, and cell ontology terms. The first step is to augment these. So we use the LLMs to expand these terms. Okay, what are these cell types in a structured description? Which pathways are active and all of that? And based on that, we use Quen models to 8 billion text model to embed it into in this case, a 8,000 element vector. And in that vector space, in that embedding space, we start running a similarity calculation. So we find the most similar cell ontology term for both predicted and author annotations. We calculate cosine distances, and then we use a kernel to score that into a 0 to 1 score of how close the author and the predicted annotations are. So just as a validation of that, probably you can ignore everything on the left. But here we are showing like we are taking the same cell type, CD4 positive alpha beta T cell, and comparing it against other cell types. And as we go down the line, these are more and more biologically more distinct cell types. And you can see the decay of cell site onto scores as they decay from one being like perfect, it's comparing itself to itself, and then decaying very quickly. And then we have different kernel weights here. It's a parameter that sort of controls how quickly these weights these scores decay as you go more and more further. And then that's where we did a little bit like we chose one of these parameters. I think it was 0.25 here, where we could see across many different cases that, we can say, okay, this is where a score of 0.5, you exit same subs major categories of cell, and then as you go down and down, you probably are not even talking about the same cell anymore. It becomes a background values. So these scores also then become a little bit more interpretable once we have these kind of decay curves for many cases. And in the paper we describe them in many different cases as well, that you can then interpret these scores. Okay, probably at value of 0.6, then we are not even talking about major cell types, similar cell types anymore, for example. So that was the whole sort of idea of using the data sets, tools to be compared, author annotations versus predicted annotations, doing it on a continuous scale using CyteOnto as a framework to compare cell types. So then we ran this across these data sets, and these are the overall scores that we got from cell type as single R and GPT cell type. And then we compared them against CyteType, but using different LLMs as the brain of CyteType. So what was the most interesting is that even very sort of small and weaker models, let's say GPT OSS, which is open weight model, these outperform single R and cell type and GPT cell type. The best performing ones were, of course, the frontier models here at that time. Claude Sonnet 4 being the in autumn being the latest model out there, and they outperformed GPT cell type by 388% and single R by almost 101%, and having much stronger results in annotation accuracy. So what you see here is the CyteOnto scores again. And then we also had other parameters that we looked at as like confidence calibration, like which LLMs, and this is where a lot of variation was. Some LLMs are really good in conveying their confidence. That means that if you're saying we are highly confident, then it's very likely that the annotation is correct. And when they say they have low confidence, then it's very likely that it's wrong. So that the most accurate method was not always the one that was conveying its confidence accurately as well. So some of them, like GPT-5, for example, was the most unreliable one. Even though it was the most accurate, it can often show strong confidence where it has produced a wrong result. So that was very interesting for us to see. And that's where we saw a lot of variability, and there was no correlation between accuracy and reliability of these models. One thing that was interesting to see, and which is actually the strong strength of our architecture that we have built, is that even if you run these models again and again, they will give you the similar answer. So they do converge onto the similar results at the end. So that means there is a high amount of determinism baked in. There is a bit of stochasticity to it, but there's still these are very reliable on reruns to be able to and perform really good there. So that was what gave us confidence, essentially allowed us to think of CyteType, that is the architecture of CyteType, of how we're doing this AI workflow, that matters more than the underlying LLM that you use. And with all the updates we have been doing, we have like really brought the differences between these models to really small delta now, and we are actually running a lot of benchmarking internally right now, to a point that you don't need to run these super expensive model. You can run like 10 times cheaper, let's say DeepSeek, and now we 3.2 models, which are 10 times cheaper, and get pretty much similar accuracy on these tools. So that's something that's a constant effort on our side to bring these cheaper models, because then you can run them on a very large scale corpuses, and that has its own value as well. So one of the last slides here was is to talk about the engineering. CyteType can consume a lot of tokens per cluster. They're now up to a million tokens, 1.5 million tokens. So imagine like when you run Claude, that means Claude has $3 per million token and $15. Claude's on $15 per output million tokens. So 1.5 million tokens can you can consume up to $4 to $5 here per cluster. So if you're processing 100 cluster, that becomes very quickly very expensive. So that is something that one needs to sort of think about, that that site type is, it produces good results, but it comes today still comes at a cost. You can run it with with the smaller models and open weight models like DeepSeek, which are more cost efficient. And of course, you then save a lot of cost there. And then that's something that we have been actively trying to do. And tool calls and structured outputs can fail. So even some of the models that are really, really high in accuracy, they fail often. That means then you have the retry mechanism baked in. That means they take longer and cost more to run. So that is also something, there's like hidden costs associated with certain LLMs. You need LLMs with really good throughput. Let's say 50 tokens per second output throughput. If they're really, and that will take around 15 minutes. If they are low and you're thinking about using some local LLMs, that you have deployed in Olama or VLLM locally, unless you have lots of NVIDIA GPUs, H100 sitting around, which will be again $35,000 each, you will not nowhere match this kind of throughput. Usually, your DeepSeek locally deployed will give you a throughput of 2 to 3 tokens per second, and then it crawls to really, really slow speeds. So that's something to also know, that if you're going to thinking about locally, you need a strong GPU infrastructure to deploy that locally and achieve, at least we would recommend, a 50 TPS speed to run it in a reasonable time frame, and 15 minutes is per cluster again. And we usually run 20 clusters at a time by default. And depending on workloads, we can even go further. But then again, you have to think about that this will scale up based on your number of clusters as well. But the good part is that local deployment can offer you privacy. And with online models, you have to think about rate limit errors, because if you're running a lot of these queries simultaneously, then you will have to think about and go to OpenAI and ask for higher rate limits as well, or Anthropic or Google for that matter. So on and so forth. Of course, at the behind the scenes, you also need a lot of distributed compute, because you'll be running a lot of computation, doing a lot of on-the-fly differential gene expression, pathway search, ligand receptor analysis as well, something that we have recently introduced to CyteType as well. So there's a lot more to think, and these are like engineering challenges that come along using LLMs. And this was one of the reasons why we actually decided to actually support CyteType by offering users, if users today run CyteType, they don't have to provide the LLM tokens. We actually absorb the cost for academics. And that's one of the good things, I would say, to be a company, that you can support that if you have like different strategic objectives. And that has allowed us to really grow SciType, learn a lot from people, their workflows, and then improve it based on the user feedback as well. So that is something how we are sort of have built it and are building it moving forward. But yeah, building scalable tools around LLMs is a challenge, and one has to really think through. And as a private company, we own that problem a little bit more than we would as an academic. And at the end, SciType produces a lot of. I will actually drop out of my slides real quick here, do a quick demo live here for you guys to get a sense of how it works actually. And of course, it's been quite a big team efforts from, again, internally from us, but also our collaborators in Lund University that have been working and helping us validate that to quite a good depth as well. So what I'll do now is actually try to see if I can drop out of my slides and quickly share my screen again here. So here I'm just went into, so you can go to our GitHub repo and you will be able to find one of these notebooks which will guide you to how to use CyteType. So here in this notebook, I'm going to do like a 30-second run through this. So we start with, for example, downloading a data from, probably zoom in a little bit more here. Downloading a data from Cell by Gene. So you can go to Cell by Gene and download an H580 files. You can quickly install CyteType and ScanPy. Fun fact, we just actually recently implemented in CyteType one of the marker gene search system, which is equivalent to ScanPy because ScanPy doesn't support reading data in back mode. So if you have a very large data set, you should read them in the back mode and data, because otherwise they're going to load up everything into your memory. But if it's stored in back mode, you can't run marker search. So that's the problem, and that's why we built a sort of a small utility tool that will allow you to run marker search in and data files even when they're in back mode. So you don't have to materialize the whole matrix upfront in your memory. And then you'll choose one of the categories. So we use one of the author provided annotations already, 24 clusters here in this case, and and this is what we will re-annotate with CyteType and see how the results look like. So we quickly on that group, we run a marker search, and then we call CyteType with that AND-data object and also tell where the marker genes are in the AND-data file and also which groups we are using for annotation. It will package a lot of your data, aggregate a lot of data behind the scenes, also create like a gene-oriented matrix and then compute that and dump that locally in your computer. And then this will be sent to our servers to be processed later. So we also do a DuckDB artifact of all your observations because that allows later agents to really on the fly look at different categories and whatnot. So runtime on this is actually not that bad. I think the packaging of data takes about, and I think in this case with 100,000 cells will take about 3 to 4 minutes, and an actual runtime can take around 20 minutes to process CyteType across, let's say in this case, 24 clusters. And once you have that, all the data is then put back into your AND-data objects. So you have CyteType annotations, you have that as an observation key, and then you will get your clusters back annotated with CyteType, and also with on cell ontology terms, the standardized cell ontology terms that they are sort of linked with. You can also, using the utility we provide, the marker dot plot, you can also very quickly just generate a marker dot plot with the marker genes that CyteType identified as like key supporting marker genes for every cluster, and also the coarse clustering of your data. So, different cells in this case, like lung epithelial cells and immune cells are all categorized into these broader groups. And then you can also have them in these nice visualizations, but then you have access to all the data in your AND-data objects as well. And then at the end, what CyteType does is that when you're running it, it will also give you a hosted report on our servers. So once you've run that, you can click on this link, and then you'll see one of these reports for your data. And in this report, you can then sort of go cluster by cluster and look at the annotation that CyteType provided, a more nuanced annotation, cell state, confidence, and then a lot of verbose information about how did CyteType arrive at what it arrived at. So you can look at what are other kind of candidates that were sort of explode, why they were explode, why they were rejected, what are what are the missing ones, what's unexpected. You don't see to that because this could be something specific about a disease state. It also runs this differential gene expression on your data against nearby cells, and then you can also enriches that information into and bakes that information into your annotation process. It tries to understand how different genes are co-expressing to understand how the heterogeneity. So it will come up with the pairs of genes and try to use that to understand if this is a heterogeneous cluster or not, or is it like some sort of co-expression of genes that's usually doesn't happen, and will provide a lot of evidence around that. And then mask away the cell type identity information and just focuses on cell state to understand what kind of active state that cell. And so in this case, these cells are activated and surfactants secreting, and then what more gene sets are supporting that is also involved. And then you try to understand the confidence. Why is it confident? Looking at all the reviewers that provided and how it's the heterogeneity of this cluster as well. We run a classifier behind the scene to see if, which other cells are sort of a confusion matrix as to which other cells could be classified into the cell type. And then, of course, you can see the detailed review from each of the reviewers and their persona as well right here, along with the marker databases they looked at. And yeah, so there's a lot more information. You can deep dive into actual pathway data analysis as well. So all of that information is sort of packaged here to be like build confidence into an annotation process, understanding the biology of that cell, something that we as bioinformaticians, don't get enough chance to dive into. And, searching through PubMed for papers and marketing is not that fun. So all the information is brought here. The most important part, because we reran it on the author's data, was that we actually can switch to and see user labels here. And then these values here will show us confidence scores. So this one here is very interesting that the authors originally mapped called hepatocytes, but cells are not hepatocytes. They're part of this very large epithelial cell sort of a supercluster, and specifically here being a lot of adenocarcinoma cells, and the system identifies as lung adenocarcinoma cells. That is well supported by the underlying metadata for these cells as well. And then you can go in and chat about it and ask the copilot as to why are these not hepatocytes, and like you can ask more specific questions. And here I did that in one of the, like, I asked it like, why are these not hepatocytes? It will go and start looking at genes. It will start looking at the tissue and disease context and run SQL queries on this, on the observational data that we had along with these cells, the metadata, and will give you a very, very detailed information about what should have happened if these were hepatocytes, if these are not hepatocytes, and will give you a sort of the exclusionary evidence, and if this is a liver gene signal contaminant artifact or what is happening there in a very detailed way. So that allows you to further probe biology and then really go in an agentic way, and let LLMs uncover that information for you. If you don't want all this information, you're just interested in the sort of the data, then you can just download the CSV file, and then you'll be able to just see the summarized information here on what these cell types are, and the coarse clustering and cell state and whatnot in the supporting market. So you can get like a summarized version of all of that directly here, and that's sort of probably more programmatically friendly. Yeah, probably I'll stop here now, and happy to take any questions that you have here.
Great, great. Thank you so much. That is really, really amazing stuff. I really want to emphasize to the audience like what a difficult problem this is that CyteType is solving. And yeah, really amazing work. Let me go ahead and allow people to unmute themselves. So I guess we have some time for maybe like one or two or like a couple of audience questions. I did want to start off with a question of my own, which is just, as you alluded to, there's a difference between running LLMs on the cloud, which may be faster, versus locally, which may be more private. So I was just wondering if you have given any thought to running this on like patient data, maybe including things like CLI, clinical information. How are you guys dealing with that?
Yep, it's a challenge, right? We have to, so for bigger pharma companies that have invested heavily into their own GPU infrastructure, it's a very easy solve for us. We actually are doing it with a couple of pharma companies, like, using their GPU infrastructure. But unfortunately, most academics don't have access to, to a DGX cluster, but we do allow, now with very small, like some people are interested in like trying like this liquid LLMs and like really small LLMs and with 5 billion parameters and really see how far they can push that. So there, the speed is good enough to try. And then what we do is we support OLAMO deployment. So if you have an OLAMO deployment, you can plug that in, and then we will route everything to your LLMs that you have decided. But then Nygen servers are still the control plane. We route a lot of information through that. But that part is really, really secure, and we have like ISO certification. We don't leak data into any third party or any LLM there. So the data comes to our servers and gets routed through your LLM, wherever that might be. So you can also do your deployments of your LLMs on, Google Vertex or, Amazon Bedrock. So you can take any open weight model, deploy it on your own instances there. And that's also another secure way that you can expose that to CyteType and provide. So we have provided all that ways to hook your own LLMs into CyteType.
Cool, cool, cool. Yeah, that is really amazing. Yeah, cool to have a local option then as well. Okay, so we have a couple of audience questions in the chat. So one question comes from Trong. Is SciTide software free to use or does it require a paid subscription?
So it's free to use. We have rate limits because otherwise we'll bleed too much money. So, but I think you can run today. You can run 3 annotations per day on your data. And I think if you sign up with your email, so that we know who you are a little bit and not completely anonymous, then we double that. So you can do up to like 6, 7 annotations per day, and all of that is free to use. So, and we have no plans of charging academics. We just want academics to use it and provide feedback and citations, hopefully.
Cool. Is that like 3 datasets or 3 clusters?
3 datasets. Yeah, 3 clusters would be too
Yeah.
small, so you won't be able to do
Cool.
anything with
That's a
that.
lot. Okay, second
And there's
question is,
there's
oh,
no restriction on number of the on number of clusters yet. We have stress tested it on dataset up to 200 clusters. So yeah. So and then performance stays quite good up to that. But yeah, there's no restriction on number of clusters. 3 datasets per day, 6 if you register with your email.
pretty crazy, pretty crazy. All right, we have a second question in the chat about multi-omics. So does SciType support only single cell sequencing, or does it also support things like RNA-seq, ATAC-seq? ChIP-seq?
Yeah, so it's only supports RNA-seq today, of course, because we are looking at genes and asking LLMs about genes, and LLMs today don't know so well about enhancers and arbitrary genomic coordinates there. But with Evo 2 and those coming along, I think there is an opportunity to do that. But not today. We have long-term sort of plans of you running like something like a scenic pipeline and then being able to support attack-seq data from more regulatory aspect with coupled RNA-seq data. We are currently building something for surface proteomics data where you have that kind of RNA like CyteType like experiments. So that's kind of on the roadmap there.
Cool, 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 cool. Yeah, it makes sense. That's more of a frontier of biology in general, right? Um, Another question comes from actually my coworker, Sophie Delbare. So, hey, Sophie. So yeah, she asked a very important question. Can CyteType perform any type of cluster resolution evaluation? For example, if I provide a macrophage cluster that actually contains different subtypes, can it recommend subclustering? Or would we need to run CyteType on different cluster resolutions and just evaluate the outcome?
Yeah, so this is one of the sort of the irritating part of CyteType. So it's not like looking upstream in your data and running your and suggesting too much about the different resolutions. What CyteType does today, if your data is under resolved or like yeah, you have like done under clustering of your data, CyteType can flag your clusters as heterogeneous so that probably you should go in and do a more deeper clustering. And it can also provide you very good evidence of what kind of subpopulations might be lurking in there. So that we have already built and extended CyteType a lot in the last few weeks, especially with co-expression analysis that CyteType does now. If you have over clustered your data, then you can also see when it's comparing with nearby genes, it will not detect any differentially expressed genes. And that is also something that we are really showing. But one has to go in and really look and go through all these data sets, which can be a bit cumbersome. So today you have to run your data set in multiple resolutions to find out because CyteType is not really about finding optimal resolution for your data. That is something we are thinking about as an extension to CyteType. But unfortunately, today you have to run it on multiple resolutions. The best way to do it is start from high resolutions and then see if CyteType starts flagging something as heterogeneous as you decrease your resolution. But the fun part is that CyteType has really, really strong performance. Let's see, and this is where it really differentiates from other tools out there. That let's say you only have T cell populations in your data or macrophage populations in your data and nothing else. Most tools will fail there because they have nothing contrastive against those populations to find out marker genes from. CyteType's contextualization actually is that's where it shows off like it's really, really, really strong, and it will be able to really flag those, different macrophage states, their polarizations, for example, and also like are they efferocytic, are they like eating up other tumor cells? All these nuances will be picked up by CyteType in your data. So I would recommend starting with a high resolution, which makes sense to you, and then taking it down from there if you feel like there is not enough resolution or not enough, like there's maybe too over interpretation of biology that's not interesting to you.
Yeah, yeah, yeah. That's great. Yeah, I really wish I had this in grad school. That would have been very useful for that particular question. So yeah, Dr. DePello, do you have time for like maybe one more question? I do see we're at 11, which I guess is 4 p.m. for you. We had one more question in the chat. Okay, so yeah, have you tested using gene activity type scores from single cell ATAC-seq?
We haven't. It would be interesting to do that, to look at the sort of a proximal promoter based activity scores or upstream enhancer based. Uh, we have, in my PhD, we have looked at like, how well do these, the activity scores translate to expression. And in some cases, especially in continuous development models like hematopoiesis, they do translate really well. So I would say that would be interesting, and I would expect it to really show good results in certain systems, but we haven't really tested it by ourselves.
Interesting, interesting. Cool. Well, yeah, with that, I do want to really thank you so much for your time. It is so amazing to learn about this awesome technique and yeah, really, really great work. Thank you to everybody for tuning in. As a quick reminder, we have an upcoming panel on April 9th related to biotech venture capital investment. So yeah, thank you very much, Dr. Parashar Dhapola, for your time. CyteType, give it a try.
Thanks, Alexander. Thank you. Thanks, everybody, for listening in.
All right, take care.